Okay, I think 5 past, so we'll kick off. Welcome, everyone. Welcome, folks in the room, folks on the call. We're up to lecture 3. Um, feels like we've covered a lot already. I don't know if it's just me. I'm surprised it's only lecture 3. Okay, so thinking about, as I'm introducing every lecture now, just the overall syllabus and the general trajectory that we're kind of following here, right? So we had our intros and talk about single neurons last week. We talked about a kind of few things related to neural networks. We talked about Hopfield networks. We talked about networks of spiking neurons, and some kind of computational properties related to pattern recognition and kind of memory type operations in neural networks. If we think about moving as we are up this, kind of in a general sense at least, up this arrow of spatial scales, last week and this week are in this area here. Welcome, guys. Are in this area here where we're on. We're talking about the activity, dynamics, computational, mathematical properties of large numbers of neurons, where large basically, we're not even going to kind of define a specific number, but we're going to say on the order of thousands to tens of thousands to maybe more of those, to the point where we're just going to describe it as like a statistical distribution eventually, and not try and put a kind of specific numbers on it. More to say about that kind of question, like finite size effects and so on as we go. But we're in this territory here. So last time we were talking about networks, kind of qua networks. This time we're also talking about networks, but we're really thinking about networks more in terms of the collective activity of groups of cells within a network. So if you like, within a small network as well. So if you like a kind of global small network or population activity properties, and what we can learn and understand about those given what we know about the elements that they're composed of. Okay, so this was last week. As I said, we kind of summarized some of these things. Some of these are going to come back in a few ways as we go, and hopefully that'll be obvious to you when they do. So as the title suggests, we're kind of focusing on Wilson-Cowan equations, but this is the kind of broader context here is this is the kind of archetypal type of mean field model in computational neuroscience. And there are a few different words or synonyms for this concept of a mean field model. Like mean field is in a way the best one because that's a term that's used consistently across a number of different scientific fields. It means slightly different things in different fields, but it's kind of broadly speaking a kind of common vocabulary. Within computational neuroscience, there are some more, say, domain-specific synonyms for mean field models. So we talk about neural mass models, neural field models, mean field models, as I said, neural population models. They're not like exactly analogous. They do mean different things, but there's this kind of Venn diagram of, say, bullet points of what you would define each of those things as, and they're kind of highly overlapping. And the Wilson-Cowan models is like, as I said, it's the same, the archetypal neural mean field model in computational neuroscience. So a lot of the kind of ideas that motivate what we'll study here for the specific model, they also motivate other models that you'll hear about from me and you'll hear about in general in the literature, that are kind of models of population level neural activity. So we'll talk about some of the kind of concepts of this, and then we get into the nuts and bolts. We'll go over the formulation, and then we'll look at some numerical and some analytic properties of these systems. They'll be somewhat reminiscent to some of the dynamical systems analysis we did in lecture 1. Which are, kind of good thing there. So we kind of reuse some concepts and reuse some familiarities that we developed before. And yeah, that's the plan for this session today. So when we were going through moving on up the scales, and in the kind of previous few lectures mentioned once or twice, this project that actually kind of wrapping up now, I think, in Europe, called the Blue Brain Project, which is not, I mean, there's lots of people doing work similar to Blue Brain over the world. It's kind of not, it's a bit thin to just refer to Blue Brain all the time, but they kind of exemplary example of detailed models of cortical columns that have all this kind of morphological cellular detail. And there was a review by Christoph Koch, who was not part of Blue Brain, and he was leading the Allen Institute at the time, but kind of reviewing the contributions and some of the observations in these detailed Blue Brain models. And there's just something that's kind of notable from this quote that that's highlighted here, which is, I'm just going to read it out. So essentially they're kind of reviewing a kind of landmark paper from this project that was doing some very, very numerically detailed computational models, which is not what we're talking about today here. But what they said was that the things that are observed, this transition, so this is just a kind of dynamical process that they observed about like moving into an oscillatory regime, is clearly describable by simple models. There's a long history with notable contributions from Wilson, Cowan, Amari, and applied mathematics, also so-called neural field theories, So like in a nutshell, the review of like the main achievements of this kind of big project doing these really big complicated models is that they've basically reinvented the wheel of things that have been understood and studied for quite a long time in the framework of these mean field models, such as the Wilson-Cowan model. So that's just a little side note to kind of motivate you there, is that there are the take-home point here is that, is what I've kind of described at the top of the slide. So there's a question of choosing the scale or the level of analysis for, sorry about that, for the neuroscientific research question at hand. So you can put together large, blue chip, blue gene level simulations. What you get out of it often is something that basically looks like a Wilson-Cowan equation. And you might as well have done a Wilson-Cowan equation, some might say, okay? So that's just a motivating point. Now, we talked a little bit about, and actually what I was referring to on the previous slide is this idea of a particular type of microanatomical structure in the brain called the cortical column, or cortical microcolumn. It looks like this in a 3D like hairball reconstruction. And these would be schematically like a few adjacent cortical columns with different layers colored, with different colors there. But the point I'm kind of making here is that we can take a very detailed representation of this this kind of feature of brain structure, brain like microanatomical wiring structure, and we can progressively simplify it. We can progressively simplify it down to kind of certain cell types receiving inputs from certain parts of the brain, like subcortical innervations and neuromodulatory projections to certain laminar locations. You can reduce it down even further. Say this is a Janssen-Ritt model sketch or wiring diagram where we just kind of summarize the cortical column with 3 cell types, or 3 populations of 3 cell types, or even simpler. And then this is actually what we're going to focus on today. So this is Wilson-Cowan. So what Wilson-Cowan does is basically, to a first approximation, says, I'm going to summarize, I'm going to describe all of this, all of the cellular intricacies of a interacting set of thousands of neurons within a cortical column, with 2 state variables saying something about all of the excitatory cells in this big population, all of the inhibitory cells in this big population, and how they interact. And that's the one-liner, thinking about what Wilson-Cowan equation's about. One of the things that's, I think, worth remembering, which we've seen already, and is a big feature with what we're going to look at today, but there's been a big feature already, is just this fact about how common the relationship between excitation inhibition is as a kind of organizational dynamical property of nervous systems and different things we can observe at different spatial scales or within different kind of parts of the brain and so on, but even down to the level of the cell, right? So I'm just kind of referring here, like we spent a while talking about Hodgkin-Huxley model. The Hodgkin-Huxley model is very much a tug of war here, kind of push and pull between excitation and inhibition, right? The sodium and potassium ion channels, and moving you over the threshold point, inducing the action potential, then kind of getting your overshoot and returning back to baseline. That comes out of a dynamical process that involves an interaction with time involved. That's kind of push and pull between excitation and inhibition. Okay, at the cellular level, or even not even the cellular level, at the kind of membrane potential patch level, right? So, and there are other contexts or other kind of structures in the nervous system where we have this that I'm not going to talk about today, right? Today we're basically talking about cortical columns. We're talking about excitatory and inhibitory interactions in something that's schematically representing a cortical column. But you have like inhibitory and excitatory nuclei, say in the basal ganglia, that have some of this push-pull relationship, and then the thalamus. So there's this, that you can see this kind of thing everywhere, basically, and you can see it everywhere outside of neuroscience as well. But it's worth reflecting, like, this is a very common principle in the neuroscience context. Excitation inhibition at the level that we're talking about here, which is between cells and between populations of cells, is, as we've mentioned before, is mediated by neurotransmitters. It's mediated by an excitation inhibitory neurotransmitters, right? So as we will mostly be talking about over the next hour, 1 to 2 hours, the kind of excitatory cells, inhibitory cells, excitation and inhibition. I just want to kind of underscore, like, this is mediated by the release of certain types of chemicals by the neurons that have a particular kind of electrochemical effect on the cells that they are kind of projected into. Okay, so that's what excitation inhibition means at the level of cells that we're talking about here. So yeah, I mentioned cortical column. There's a lot to say about EI interactions. So this little term I've set at the top here, EI interactions, excitatory inhibitory interactions. This needs to be in your neuroscience vocabulary. Like, you'll see this written on like every other paper you read, to be honest. Like, I certainly do. It's a bit of a catch-all term, but I mean, it's a catch-all term that says something along the lines of point I was making on previous slides, which is there's interesting things about how your excitation inhibitory processes are that extend beyond some of the simple examples I'm going to show you today. So there's a number of them summarized in this paper by Thiele Wallemster from a few years ago. Things like feed-forward inhibition, right? When you have excitation onto an inhibitory cell, that's in this case, like we say, thalamus kind of driving this guy, parvalbumin positive cell, that is then inhibiting this guy, right? So we call this feed-forward inhibition when you have an inhibitory effect on something by having an excitatory effect on something that then has an inhibitory effect on that second process. And often with feed-forward inhibition, it becomes a question of balance between the kind of tonic level of innervation here versus what your kind of modulating effect is, whether you kind of flip the balance of the inhibition on here so that it is having a net inhibitory effect or is having a suppressed inhibitory effect. And there's a lot of kind of nuance to how that can behave in different circuit types. Other examples, I'm just kind of showing a few examples just to say that, again, like the EI interactions are going on in a lot of different contexts. So this is a canonical variant of more or less what I think we could think about the Wilson-Cowan model being, which is this just EI circuit within two primary cell types within a cortical column, the pyramidal cells and one of the primary inhibitory cell types, the PV cells. So this just two population interaction with this inhibiting this exciting this, right? This is a kind of what we call ping mechanism. There are non-chemical or non-chemical neurotransmitter types of inhibition, which I've mentioned previously, but I don't want to elaborate on because it gets complicated. There's a lot of other, say, variants of these, even just 2, 3, 4 component interactions that have been studied to death in neuroscience. So, you can find a lot of richness around the question of what EI interactions can generate in terms of, behavior of neurons and neuron populations. Yeah. So in this review from Thilo, they're actually associating these different motifs with frequency ranges. So they're kind of saying, yeah. So it's, for example, like, and this is, I think, quite well established, is that this ping mechanism is one of it is probably what's driving gamma frequency activity that you see in LFPs. Yeah. Yeah, that's what these diagrams are kind of getting at. I mean, I think to be honest, that's what they're saying here. I think if you kind of look elsewhere, you might find people saying something, different. I don't think it's clear cut what the dotted lines. And you mean here? Well, I'm not really, I don't remember off the top of my head what the point of the dotted lines here. I mean, the usual, I'm sure you're familiar with the usual, diagram here. We've got like a flat line here saying we've got inhibition. Inhibition by S. Yeah, I'm not actually sure what the point's making with the dotted lines here. Oh, it like cuts it out. Okay, yeah, that would make sense. Although we're not getting that here. So I think maybe, yeah, if the point will be like, if this inhibits this, then that cuts out this inhibitory connection on this. This one? No, on the inhibition this one? Yeah, that one. Yeah. One of the ones coming out of PC and the other one. So, well, if this is turning off this, then this is no longer exciting this, and maybe this is turning this on. So when this is not turning this on, then this gets turned off. There's like a sequence of things being turned off by presence of this. I think it's a good question. I think it's probably, I expect it's well described in the legend. I'm not too concerned to kind of give the bottom line on it right now, but there are kind of circuit switching structures that have been studied a lot in specific characterized neural circuits in cortical columns and other types of structures that are, and also have interesting kind of spectral features as well. But I think, yeah, like ping gamma is I think the one to really internalize here. This is what everyone knows about, and I think is quite well accepted. The others, they're a little bit more speculative, but don't hold me to that. Okay, so we'll get on to the main item here. So this paper from 1972 is the paper that the Wilson-Cowan equations are, I guess, named after. the history to this. There's actually what I want to recommend is there's a few articles by the senior author here is Jack Cowan. Okay, so he's kind of I guess the brains behind this collaboration, because I think Hugh Wilson was a postdoc at the time. But you can see in some reviews that Jack Cowan's written, and also there's one or two like pretty nice lectures on YouTube where he kind of describes the kind of what led to the development of this model. So to kind of summarize a few of the points from that is the question that they're going with here is what is how does the kind of interplay between excitation and inhibition manifest in neural dynamics at this population scale, right? Population scale being localized populations of neurons, right? So I was talking about these as a course called Column, being a canonical example of that. So the general approach here that is kind of emphasized through this paper is there's kind of two main elements to it. One is the, let's say, kind of conceptualization of the problem of describing a population of neurons as a mean field kind of phenomenon. And then the other part of it is the dynamical systems analysis of what we get from it, which is a two-dimensional dynamical system. And that follows some of the structures and some of the kind of approaches that we saw previously with the other two-dimensional models that we studied. But this work is often credited with kind of really introducing things like phase plane analysis into mathematical computational neuroscience, or like popularizing it, essentially. I think they weren't first to do it, but they maybe it really kind of gave it a boost in terms of it being a commonly used technique. So the idea is this. So saying we're talking about neural populations or neural ensembles. Okay, so the innovation here, or the kind of difference relative to, say, talking about single cells, is that we're talking about now populations of cells, large number of cells. And the reason for this is just general understanding that firstly, a lot of the things we measure in neural recordings, because a lot of the time we're measuring, if we're doing non-invasive, say, scalp level recordings, we're necessarily measuring the activity of large numbers of cells. Even if we're in the brain with like a depth electrode during surgery, we have local field potential recordings, we're still measuring hundreds, thousands of cells and the kind of extracellular effects of the kind of membrane potential summations of currents and so on. So that's kind of even that is like a mean field level kind of data quantity. So the only time when you're not looking at something that's like a mean field type like data quantity is when you have a cell on a dish and you have a very, very small electrode that you're able to kind of stick it inside the cell and not break the cell. So you can measure, and you can do a patch clamp, for example, and you can measure something intracellularly, which is a lot of neuroscience, but it's not, you kind of miss a lot if you just study isolated cells. Let's go with that. So the approach is to actually go with a relatively simple type of description of the cell of these two types of cells. We're going to talk about excitatory cells and inhibitory cells, right? And if you recall, that definition of excitatory cell is a cell that releases an excitatory neurotransmitter, and vice versa with an inhibitory cell. And then the thing that we're going to be tracking is the proportion of them that are active. Okay, so this is a simple kind of heuristic idea of how to think about the behavior of neurons as being like they're either on or they're off. They're active or they're inactive. Now that's it's kind of obvious, but it's, oh, these guys were aware of it as well. Like that's a simplified schematic description of a neuron. Like neurons aren't just on and off. They fire, they have, temporal dynamics in the activation of their membrane potentials and so on. But it's not a bad summary of them either. Like they're fire, particularly if we reduce it down to like firing and not firing, with some additional say recovery dynamics that we'll get to in a bit. So that's kind of the starting point. We're going to talk about proportions of active cells, proportion of inactive cells. And the basic question is how do those go up and down? How do the proportion of active cells go up and down? And how's the proportion of active excitatory cells go up and down? How's the proportion of active inhibitory cells go up and down? So this is the kind of diagram schematic, as I said. First, I want to talk about these two pieces, the E and the I, and then I want to talk about the way in which they're connected together. Now, you'll remember with the Hopfield networks, we had something similar, right? We were talking about elements that could be on and off, and then they were kind of triggered to become on if the input crossed a certain threshold value, and that threshold was defined by a sigmoid. So it's pretty much exactly the same thing in the case of Wilson-Cowan. So I'm kind of representing that here with on, off, on, E on, I on, and quiescent, I'd say. But bear in mind, if you kind of use this figure directly, compare it to the Hopfield network, these were the this was the connection matrix rather than the cells. So this is, but here I'm just kind of schematically saying cells are going to move between being active, inactive, active E, active I, and quiescent. Okay. So we'll define these quantities. We'll say we've got N cells where we've got like NE and NI numbers of cells in each type. And then we're going to define a proportion, which is the total minus, so it's the proportion of active cells, active E, active I, and then the proportion of inactive is just the total number minus these guys, okay? So there's always going to be kind of a symmetry or a complementarity between the proportion of the inactive and the proportion of the active, okay? And we can think about these quantities, E and I, as neural masses, right? Again, that's the one like term of art in computational neuroscience for mean field representations of neural populations. So we're going to have these quantities. They're going to behave like kind of Poisson processes in the sense they'll have a Poisson distribution on the spike timing or the firing timing, the interval between firing. The important thing is they're going to move between an active state or an inactive state, and that movement we're going to summarize by an arrow going up or going down. So like so. So we'll have a probability for a transition probability of a certain neuron going from active to quiescent. So the down arrow here for a certain period of time, say dt, going to be this defined by this this constant, and this is our basically a decay constant, so decay rate. So that's for our decay rate of an active cell, and then we're going to have the same thing. We're going to have a transition probability for a neuron going from inactive to active, from quiescent to active. So up arrow, and this is again going to be defined by a transition constant. In this case, this is the maximum firing rate, so we call this constant. And in this case, we've also got a function on of an input function. So this is different from this, right? And the point here is that in this case, we've got some cells that are active. They're active. They're going to decay back down, kind of just as a natural decay process back to the resting state, which is quiescent. We want to move them from resting or inactive to active, we need to have some push. And that push is going to be coming from whatever is like on the right-hand side of our activation rate constant here. And it's going to be basically some input that's passed through a sigmoid is how to think about this. Okay. So as I was saying just there, so our activation function, like we had with Hopfield, like we have in artificial neural networks, like we have to some extent in single neurons with the FI curve. FI curves don't look exactly like this, but it's got a sigmoid that's going to tell us, okay, if our input level is at a certain amount, then we're going to have either not much, and then we're going to have some like linear domain where we have a certain probability of moving from inactive to active, but that's going to cap out at the saturating part of the sigmoid. Okay. So with that as the setup, we're going to think about the flux over an interval where we have a transition in the number of cells that are activating minus the number of cells that are moving into activation minus the number of cells that are moving from activation to inactivation, and then the number of E cells, the number of I cells that are doing that transition over a certain time interval. Now, question on the chat. Hi, regarding total populations, E and T are proportions. If so, do we visualize them to be multiplies then? Okay. Hi, Ravindu. Thanks for the question. I think I'm going to keep going as I usually do. I'm going to keep going for a bit, and then I come back to your question. But we see it, and I'll make sure I get back to it and answer that question in a little bit. Okay. Okay, so we have number of cells becoming active is going to be proportional to the number of cells in the network multiplied by these transition probabilities. Okay, so this is our activating transition probability, and what this is basically giving us here. So this quantity multiply this is basically if we have if this guy is large, so if we have a large number of cells that are currently firing, there's not very many that are available to transition. Okay. So the kind of magnitude of the transition rate or the transition number is going to be a function of the well, it's going to be larger. It's going to be larger when there's less available, and it's going to be smaller when there's more available. And what's going on here is that one way to think about what's going on here is that the kind of neuron number, the network size, is giving a kind of upper limit on the magnitude of what these terms can become. So it's kind of playing a kind of thresholding role. Side note, I mentioned a little bit, or I started talking, I think I got distracted, but I started talking about what the background was for the formulation of these equations. So there's a few pieces to it, but one interesting one is the influence coming in from population ecology. So one of the kind of celebrated systems, mathematical models in ecology is called the Lotka-Volterra equation, which is foxes and rabbits, I think, is the kind of canonical example of that. So you have predator-prey, right? So you have a certain number of predators in an ecosystem, a certain number of prey in an ecosystem, and then the more predators there are, the less prey there are because they're eating the prey. And then there's like a lag time where if there's there's not enough prey, then there's going to be like the predators will die. That's a little bit different than in the neuron case, but you get this kind of structure that can lead to oscillations where you have the magnitude of basically your equivalent of your E variable and the magnitude of your I variable, they kind of flip-flop, and they're structured by like the number of kind of available spots in the population. In the ecology context, that's called the carrying capacity. In this context, the analog of the carrying capacity is the neuron size, the number of neurons in the population. So that is the carrying capacity of the E variable, like the carrying capacity of the proportion of cells that can be active is the total number of cells. Okay, so, and Jack Cowan was kind of influenced by this work that he'd read about the kind of dynamics of ecology networks and excitation inhibitory kind of processes effectively that have been studied in certain kind of ecosystem models. So we've got some flux in the proportion of our active cells that's related to the number that are available to transition. Same for both the E and the I. And we have our as I mentioned before, so we have this input variable, this activation rate variable here, which is going to say that the transition from the transition into the active state is going to be also driven by a kind of input amplitude that's going to be a function of the S term that we'll talk about in a minute. So this for E, this for I. Okay. There's some stuff. Yeah, these next two slides. There's some stuff in these slides that I'm gonna kind of not go into the details of here. That is talking about this in terms of Poisson, kind of stochastic Poisson equations. But I want to kind of move to this and pick it up from here. So so what we're going to go to is an equation for the evolution of our state variable that is going to look like so, where we have our activation, our decay rate on our E, like we had before, our activation rate, our maximum firing rate, and our 1 minus kind of. So this is now kind of normalized in effect, or rather we've taken n up to infinity. So now we don't have n minus, we're just doing 1 minus. So we're saying that this is effectively like some fluctuations around a normal distribution, rather some fluctuations around some quantified size of the population that we're going to put a specific number on. And this is usually what we're talking about and what people are thinking about with Wilson-Cowan equations. They're not trying to say this is how many cells are active in this population. They're really trying to say this is what the average activity level is, and we're going to give it some this kind of arbitrary or normalized unit type value. Okay, so this is the kind of component of the Wilson-Cowan equation that's going to tell us about the kind of rise and fall of the proportion of active cells and the proportion of inactive cells, just due to the kind of internal dynamics of the E and the I populations. The next step is to connect them, because at the moment we're basically saying we've got a population, we've got some E cells, we've got some I cells, they're not interacting. Okay, there's no DI anywhere in here, or there's no I. Okay, so that's the next step. And thinking about this relates to what we talked about previously with thinking about the inputs to a neuron within a network. It's going to be we have some presynaptic population, postsynaptic cell. Influence the presynaptic population on the postsynaptic cell is a function is scaled by the synaptic weight, and then that synaptic weight could be signed such that it's positive for excitatory cells and negative for inhibitory cells. And as I've said, we're kind of normalizing things by the magnitude, so we're going to get a kind of unit normalized weight quantity. But this, so we've got some weights that are coming in from all of the other cells, and we want to summarize that. We want to give a kind of summary variable of effectively, or some like an aggregate number inputs from basically these cells and these cells, from all of the E cells and all of the I cells onto all of the E cells and all of the I cells. So it's kind of, we introduce these quantities here. So W, so WE, WEI, WIE, and WII. Okay. And those are going to come inside our sigmoid function. So this was what we had up to here was this, and then we had an S inside here. Okay, S inside the F. So now we're kind of just elaborating on what the contents of that S inside of the kind of activation function is now. And specifically, we're going to say there's some input from the inhibitor, from the other cells in the network. There's two sets of inputs from the other cells in the network. So we've got some inputs to the excitatory population from the excitatory population, and we'll scale those by this parameter W sub EE. Okay, so self connections. I don't have the diagram here, but okay, if you recall the diagram. Okay, I'll just quickly skip back to the diagram if you want it. Okay, call it J here. We're calling them W in the slide down the road. So the W's that we've got there are going to be WEE for a self connection on E, WII for a self connection on I, okay, in this case, WIE and WEI for E to I and I to E. I'll just comment here, this kind of notation where the order that you choose for your indices, indicating whether it's like from A to B or from B to A, you'll see either convention for that. So you can see here, like this is in the convention used here is saying E to I is being called IE, and I to E is being called EI. Sometimes you'll see E to I defined as IE. Sometimes you'll see E to I defined as EI. I'm not sure if there's like a universal correct accepted notation for that kind of thing, but just be warned. Okay. But you know, the important thing is just define the terms that you want to use. So here, back to where we are. All right. So this is the full, say, like canonical Wilson-Cowan equation here. We've got it, we've kind of added in all the pieces. We're just kind of working through the pieces now. And yeah, the notation here is the opposite of the notation I just showed you, kind of making the point I just made. So as I said, we've got our self connection on E. So saying that, I'm just going to back up. So what we're saying here for the terms kind of from beta onwards is the evolution of E is going to be driven by the kind of decay rate and then the activation rate as a proportion of the number of neurons that are available for activation, driven by and then scaled by some input amount. And the input amount is the kind of activation level to the population. Okay. And the activation level to the population in this case has three components. It has a component that's a self feedback from our E. And then it has a component that's coming from our I, and it has a component that's not coming from the E or the I. It's an external input. Okay. So exactly the same thing on I, like without any difference at all. Well, I mean, kind of just swap these around, but as you can see, so then we've got, you know, decay rate, and then we're going to say, okay, now the input driving to the I is basically whatever the weight is from the E population to the I population, multiply the value of E, and then whatever the self connection weight is from the I population, multiply the value of I, and then we'll have some external input there as well. Okay, so These are full canonical Wilson-Cowan equations. This is what we're going to be playing around with in the tutorial later. Second comment on this slide is about interpretation and kind of variance of how these quantities are understood, essentially. It's a bit of a subtle point, I suppose. So here, and everything I've told you so far, the quantities that we've been talking about, so the evolution of these state variables, E and I, the evolution of a state variable is proportion of cells that are active, okay? Proportion of excitatory cells that are active and proportion of inhibitory cells that are active, rate of change of those quantities. And that's how it was introduced and conceptualized in the original paper in 1972. Fast forward a few decades, that there's essentially the Wilson-Cowan equations are, as in like something that has almost exactly the same structure as these. You'll notice differences here that I'll tell you about in a second. Are often not actually thought of or interpreted as the proportion of cells per proportion of cells in the population that are active, but they're actually thought of as firing rates, population level firing rates. So that's something that is more of a, say, direct analog to an individual cell. Okay, so a rate model of a neuron would be a model of a neuron that describes the activity of a variable that is the firing rate. Okay, as opposed an alternative model of a neuron, say like a conductance-based model of a neuron, will give us the rate of change of a state variable that would be, say, the membrane potential. Okay. But the kind of interpretation of Wilson-Cowan equations as rate models, specifically as firing rate models, would be, if you like, thinking about the population level activity as kind of a close kind of analog, or just kind of transferring your understanding of how you think about firing rate at a neuron level. So we're talking about a population average firing rate. And the fact is that like the maths and the equations basically end up being exactly the same. So and it's quite because we've already kind of blurred quite a few things in terms of the physiology, in terms of the details, then it's really boils down to kind of what label you want to put on the terms in the equation. But the terms in the equation won't really change whether you think about them as rates or you think about them as population averages, broadly speaking. Now, the exception to that is actually this 1 minus, right? So sometimes you'll see versions of the Wilson-Cowan equations that are giving you, that we've got at the top that I talked about before, where we explicitly have this this first pair of terms here, where we've got activation on E and then 1 minus E multiplying the rest of it. Sometimes you just won't see the 1 minus E. You'll just see activation on E and then plus this. So you won't get this coefficient in front of the input term, essentially. And so the difference there, and that's what this example at the bottom is. So the difference there is effectively the way in which this coefficient controls saturation. So again, I mentioned it before, right? Like this can't go over a certain amount because it is just balanced by the fact that the sum of E and 1 minus E has to equal 1. So that would be the kind of proportion of cells version of the interpretation equations. The firing rate interpretation equations doesn't kind of bother with this coefficient, and the reason why that works is because there's some other saturating quantities in the system that kind of stop it exploding. Specifically, there's the sigmoids. So the sigmoids actually, sigmoid here was is G our sigmoid on this one? Yeah, G will be for some reason, in this example, we're using G as a sigmoid, which is not a common choice for the symbol there. But anyway, you can see like you just kind of drop this and then do some kind of translations, then these are basically the same set of equations. But this is now talking about a firing rate version of Wilson-Cowan equations. Some other kind of fun things in the slides that I'm not going to go into is the bit that I skipped basically is just explaining a little bit that there are different, let's put it this way, like there are different approaches you can take to arriving at the same system of equations in the Wilson-Cowan case. What we were talking about before was, actually, I've got the reference for this on the next slide, was partly coming from this review paper from, well, not review paper, this paper from Wallace and Co., which Jack Cowan is the senior author on, that's kind of exploring a stochastic kind of extension of the Wilson-Cowan equations, which was not doing exactly the same method for the derivation as in the original paper, but there are different ways of arriving at the same set of equations. That's the bottom line. Okay, so that was a lot of maths without very much, I guess, neuroscience, or certainly not very many pictures. So let's just summarize some of the things that we were talking about just now. So actually, I think I had this figure in the first, maybe I had this figure in the first lecture, or I had a similar figure that we looked at in the first tutorial as well. We're just going to move back and forth between these. So, you know, what is the thing that we're talking about when we're talking about these mean field models? Well, they're the mean of the activity of the population. So a nice way of representing that is actually having a spiking neuron model for a certain number of cells, like a couple of thousand cells, and the average of that quantity, and the equivalent mean field, the Wilson-Cowan equation kind of summary, simplified version of that system. So this is what we're kind of showing in this and the next slide here. So I just want to show, so we've got this raster plot. We've seen raster plots a few times already, where we've got number of cells. The trace here is the average. And then between this and our next slide, we're kind of moving between a asynchronous and a synchronous state, okay? A level of high and low synchrony. The high synchrony being the kind of the coherence of these lines on the raster plot. But the Wilson-Cowan equation is just giving us the fluctuations on the mean here, and what we can see in this figure on the right-hand side is the power spectrum and the kind of presence of oscillations at certain frequencies. And this is an oscillation that's a kind of gamma frequency oscillation. As I mentioned with the ping model, that's mostly when people are looking at Wilson-Cowan models as models of oscillatory activity in cortex. It's something along the lines of a gamma frequency rhythm, so something like a 30 to 80 hertz rhythm, that's typically something you measure with an LFP, not so much with scalp EEG. Now, yep. Okay, another question. Is there any random terms used in these equations? Sharon, okay. Sharon, I'll answer your question, and Ravindu, I'll get to your question as well before I move on. But I want to finish up on this slide. So we talked a bit in the first lecture about phase plane analysis. We're going to see more of this, and we're going to see some of this in the tutorial as well. But just to kind of finish off what we were looking at here, so that the phase plane analysis kind of approach with Wilson-Cowan is really simple in a way, because we have these E and I state variables, so we just plot them against each other, kind of obvious. And all of the tools that we've thought about a little bit in the previous lectures about studying the fixed points and studying the kind of dynamical structure of these two-dimensional dynamical systems are to hand for understanding things like the emergence of these oscillatory peaks and these rhythmic patterns in our Wilson counterequations. We're going to be looking at some of those a little bit more next. Okay, so these two questions. So Ravindu, you were saying, good afternoon, Dr. Griffiths. Regarding the total population N, both E and I, how is that known? You mentioned ET and IT are proportions. If so, do we visualize them to be multiples of N? Maybe I'm understanding this wrong. Yeah. So I think I mentioned, you said, how is that known? As in, like, how, I think you're saying, Ravindu, how is the value of N known?
Yeah. Hi, Professor. So yeah, like how do we know this quantity? And earlier in the previous slides, you mentioned infinite number of neurons as well.
Yeah.
So I just want some clarification on that.
Yeah, so the end point is to kind of drop N, right? Because generally, like, we don't know N. Certainly, like, in an experimental context, we don't know N. We might be able to sometimes put a kind of guess on N, but it's as in the number of neurons. But usually, we kind of don't really even want to be worrying about that when we're working with these type of Wilson-Cowan equations. So N, when N is used and people are like thinking about N like over and above just the derivation, it's there's some phenomena called finite size effects. The question, broadly speaking, is when does something like a mean field, which has some Gaussian assumptions that I'm not going into here, but kind of assuming that the mean of the population is a good representation of the population, what is the size of the population in which that becomes valid? So those, and Jack Cowan's done a lot of that work, I think, over the last 20 years or so. So you'll see, if you look at some theoretical work on Wilson-Cowan equations, looking at things called finite size effects, you'll see N kind of featuring that as a kind of relevant quantity, because they want to see, like, what is the value of N where this works and where this kind of breaks down. But that's still all in models, and it's kind of a theoretical exercise. So again, to your first question, like, how is that known in relation to, say, a particular like recording or a particular kind of piece of data that you have? That's usually not the intention with Wilson-Cowan equations, to kind of say be giving a concrete number for that. The intention is to kind of continue with the derivation to the point where we are talking about something that's kind of a rescaled normalized quantity. Okay, does that answer your question?
Yes, Professor. Thank you very much.
Good. And then Sharon says, are there any random terms? So yeah, I think okay. Sharon, what I was skipping over when I was moving through is a few slides in the middle of this, which I'm going to leave in, but I'm not going to kind of go into now because it'd take too long, and I'm not actually very good at going through the specific parts of the derivation. But essentially, if you look at the paper here, this Wallace paper, which is a PLOS CB paper, so it's free, then you'll see the version of the Wilson-Cowan equations that they're looking at there are a version that has, trying to find like the summary term here. Like it's a stochastic version of Wilson-Cowan equations that has like the deterministic part and the stochastic part. The deterministic part is the bit that I've been talking about. The stochastic part is going to be like plus Ornstein-Uhlenbeck process that is just going to be following some like simple Langevin equation. So, and then the thing that's of interest when in the stochastic versions of the system is noise, exactly. But there's a few kind of phenomena where things behave differently in the deterministic than in the stochastic case. For example, things like the cycle skipping, or that you don't need as much strong coupling to generate some of the features that we kind of want to replicate in the stochastic case, and that is probably important, right? So that I think there's a few things that are like relevant when talking about noise, and another is just like what is the baseline behavior, what is the kind of say spectrum of the system at a fixed point when it's not in a limit cycle. Okay, so if we have noise in the system there, then depending on the amplitude of noise, we can generate some structured like quasi periodic activity that will have a peak in the power spectrum and generate some interesting dynamics, but it's not kind of locked into a limit cycle regime in the phase plane. So noise is definitely important there as well. But I'm kind of skipping noise here, but there's definitely, if you check out, for example, this Wallace paper, then there's a lot of interesting work on kind of stochastic extensions of the core Wilson-Cowan equations. Hopefully that answers your question. Okay, so now we want to talk about a little bit of the dynamical systems analysis. Okay. Any other questions? All right. So as we saw with, I think we did this on the Morris-Lecar equations in lecture 1, kind of standard approach for doing a linear stability analysis on this is we can say, okay, we're gonna set our derivatives to 0, gonna find some set our derivatives to 0. That'll give us the definition of the nullclines, our E and I nullclines. And then we're going to choose a point where our nullclines intersect. That's our fixed point. And then we're going to kind of focus on what is the behavior of the system around that fixed point. And we're going to linearize it as well. So when we don't have our noise term, then the equation for the definition of the fixed point is going to look like this. And then what we can do is just assume that around the fixed points we've got just small actually Gaussian fluctuations that are linear. And then with that assumption, we can proceed with doing a Taylor expansion around the fixed point. This Taylor expansion is actually has the deterministic and the stochastic part that we're not, sorry, that's not correct. So we're going to do a Taylor expansion. We're going to say, give me the kind of first, second, third order, higher order terms, telling us about the partial derivatives of the system defined by the Jacobian here. So this is kind of telling us how our variables move in relation to each other. after our linearization. I'll pop that in this matrix A, and then we want to calculate the eigenvalues of this matrix A, the Jacobian. And this, so the eigenvalues of Jacobian basically going to give us some complex valued numbers. They'll have a real and imaginary part. The real part is going to tell us about stability. Imaginary part is going to tell us about frequency. So if the real value of the eigenvalues of the Jacobian are negative, then we're gonna say this is stable. If the real component of the eigenvalues are positive, we're going to say it's unstable. And if they're kind of somewhere around zero within some degree of error, we'll call this metastable. Okay. So that's stability. And then the second part of the eigenvalues of the Jacobian is the imaginary parts. The imaginary part is going to give us the linear frequency associated with the fixed point that we choose. Okay. And then geometrically, we can kind of go to our phase plane again and think about just the kind of geometric dynamical structure of those scenarios where we've got zero frequency, so no oscillatory activity, non-zero frequency, so I've got some oscillatory activity, so this one are all omega. Our imaginary component is non-zero, so we have some periodicity, and then the difference is whether the real part is kind of expanding, contracting, or neither expanding nor contracting, going to give us a unstable spiral. Okay, when this is greater than zero, so we're kind of moving, we're expanding, kind of exploding outwards from a stable fixed point. Sorry, from a focus, or the converse. When we have a stable fixed point, we do have some imaginary part, and we're kind of spiraling in. And then in this case, this will be our limit cycle. Okay, so that's the kind of general approach for linear stability analysis. We're going to be looking at this in the tutorial. We can do some other work that I don't want to go into here about defining an analytic spectrum. But the general structure of that with the Wilson-Cowan model, again, is going to be where we can think about the spectrum. I'm just focus on this one on the left here now. Focus on the spectrum and see when we kind of vary the parameters, in this case, like the connection weights, how does that change the frequency peak, the frequency amplitude, maybe the presence of harmonics, and some other features like the kind of decay of the 1/f. These are all properties of the analytic spectrum that are kind of interest when we're looking at oscillatory patterns that we measure in neural tissue, and we can replicate a lot of the phenomenology of the power spectrum from the linearized version of the Wilson-Cowan system, linearized around the fixed point, and then simplified to the kind of analytic spectrum equation. But we can also just simulate and look at the power spectrum numerically. So last two slides, and then we're done on the first part of the lecture, and then we can take a break and go on to tutorial. Why Wilson-Cowan equations useful? Okay, so I mean, broadly speaking, like what do people do with Wilson-Cowan equations apart from kind of slightly navel-gazing mathematics? Shouldn't say that, Field Institute, right? So the big one is gamma. Okay, so like most of the work on, I would say most of the solid physiology work with Wilson-Cowan equations is doing some variant of looking at say, noisy or recurrent oscillations that are in this gamma frequency range. Gamma frequency usually defines something like 30 to 80 or 30 to 120 hertz. In neural recordings that are kind of mesoscale. So usually that's going to be a local field potential that you can say in humans. This is something you measure with a stereotactic EEG, kind of implanted depth electrode. In mice, there's all kinds of ways of measuring this kind of activity. Optical methods are probably the best at this point. But not, I think the key point is like, you don't see much gamma activity at the kind of macro scale. So we don't really see, if it's about human EEG, we don't see much kind of strongly periodic gamma frequency activity. It's just not really visible at the kind of centimeter scale. It's something that you can see at the millimeter scale. So that's where the kind of hotspot is for Wilson-Cowan equations, I would say, is like gamma frequency activity in kind of mesoscale LFP recordings. And the kind of lining, doing the same kind of thing we were talking about before. So like taking some rasterogram, trying to kind of model the population average, and maybe also looking at what the alignment is between spikes and the population average in some model of the gamma frequency activity. Another extension of that is actually sensory, somatosensory, particularly visually induced gamma oscillations. So this is something that is quite a robust experimental phenomenon. So there has been a lot of work on this, I think a good amount of it actually done by Hugh Wilson, who, by the way, is actually in Toronto. He's a emeritus professor at York, I believe, and a long-time associate of Fields Institute. And yeah, so Hugh Wilson's done a lot of work on kind of Wilson-Cowan models, specifically look as a tool for studying gamma frequency oscillations in the visual cortex induced by typically these type of stimuli where you have some kind of grating, and then the frequency of the grating or the kind of contrast level of the grating will induce a certain, and they're flickering as well, so they're kind of flickering between black and white, will induce a strong effect in, this will be say one of these recording points. So this frequency over time, and this kind of big blob here is like a gamma frequency response to a flickering grating at a certain frequency. And then there are kind of different aspects to the frequency structure of these responses. There's also a spatial component, things called that kind of relate to like pinwheels in the visual cortex, people might have seen. So kind of parts of the visual cortex that have a retinotopic organization. There's been, I think, a decent amount of interesting work using Wilson-Cowan models to kind of study that, also extending that to the actual lateral spatial structure. So neural field Wilson-Cowan equations, which is something we'll talk about in the next lecture. And yeah, I mean, there's others we could talk about, but those are my two kind of highlights on what I think is some of the more interesting work in applying these to try and understand neural systems. I'm going to wrap up, and I see there's another question there, so I'll get to Ravindu's next question in a second. So in summary, as I said, we've got like excitatory interactions. You know, you can forget about Wilson-Cowan equations, but that's something that is going on in the brain in a lot of different ways. And as I said, outside of the brain as well, right? Like the Lefebvre example that kind of motivated the formulation of Wilson-Cowan, that's like an EI interaction in ecology. So this is just a general property of dynamical systems, but they really kind of come into force in computational neuroscience in systems like Wilson-Cowan equations. So these are one of the first to really emphasize this, particularly as a driving factor at the neural population level. Wilson-Cowan is, still, and I don't think it's ever going to stop being, like, the archetypal mean field model. It kind of, it's a bit odd, I would say, that if you remember, I was talking about sometimes we interpret the quantities as like rates. Sometimes we can interpret them as proportion of cells. Yeah, I mean, you can kind of move quite smoothly between those two interpretations of the variables, slight differences in the equations. So the one that is probably the archetypal mean field model would be the version of the Wilson-Cowan equations that is the rate one. So it's like a population firing rate rather than the proportion of active cells. That's the proportion of active cells bit is really kind of quite specific to Wilson-Cowan. I don't think it's really something that is picked up in other types of modeling approaches. But yeah, Wilson-Cowan as a rate model in general is definitely archetypal mean field model in computational neuroscience. And as we saw a little bit there, and as I said, we'll see a bit more in the tutorial, we can kind of use these two-dimensional phase plane analyses and dynamical systems concepts to kind of study things like emergence of oscillations and the kind of bifurcation structure that come with variation in the parameters, and understanding the properties of these systems. And there we go. Okay. So have a think about any questions in the room. I'm going to see on the chat here, we've got Shivank says, the phase portraits were for the differential equations, right? Yes, but well, there was a schematic phase portrait, and then there was some, let's see. So this is just a general diagram, Shivank, but we'll look at some actual phase portraits generated by these equations in the Python code in a few minutes. But this figure here is just a schematic about like phase portrait types for different versions of your Jacobian eigenvalue values. Okay, Shirin says, wow, that looked and sounded so much like Riemann zeta zeros behavior. Riemann zeta zeros. I'll be delighted to learn what Riemann zeta zeros are. I actually don't know, so I'm looking forward to learning that from you, Shirin. Okay, Ravindu, for the case of metastability, is it the same concept as saying half stable? Even so, could we do some center manifold reduction of the system to approximate the behavior a bit better, or is this concept of metastability well established based on prior studies? Right. Okay. Could do some center manifold reduction to approximate behavior a bit better. So I think I believe it's the case, Ravindu, that, but I might be wrong, that there are a few variants of definitions of metastability knocking around in dynamical systems, certainly in dynamical systems of neuroscience. This is a very concrete one that is, obviously it's kind of a condition on the kind of value of a certain quantity, of an eigenvalue quantity. Another definition of metastability that you'll see is kind of whether a system moves between attractors but doesn't sit on any of them. So that's something that's, I think, more of a kind of qualitative description of metastability that's to do with trajectories within an attractor landscape. I think there are other definitions of metastability as well. I think there's a version of that in the the oscillatory model that I'm blanking on the name of right now. So yeah, I think concept of metastability is something that, I mean, the general concept is what you're getting at there is you're saying half stable. Yeah, it's like in between stable and unstable, right? That's the gist. I think what you'll find is that there are a few different kind of realizations of that idea across numerical and empirical systems, and what's the kind of concept with the most currency depends on what people have been thinking about mostly within that specific domain. Question. Yeah.
Like especially just in the theory of differential equations,
Yeah.
after you linearize a system like that, those are the only options. So like you
Yeah.
try and change any one of the signs of the eigenvalues in that case, like you're just gonna fall into a stable, unstable, metastable. So maybe like other than just like an oscillatory, like cyclical pattern, the only other like metastable option you can think of would be some really weird, like almost you can imagine like an orbit through space, like from the different equilibriums being pulled from these other ones, but the likelihood that that doesn't eventually converge to another equilibrium, like you'd have to like spend a lot of time to try and like create a linearized eigensystem that would be able to kind of pull and push away from all these different eigen solutions, but never converge.
Yeah,
So
yeah.
metastable definitions, well, I know probably one of the strongest.
Yeah, I think you're exactly right. I mean, in the restricted context of a system where you can define some eigenvalues of a Jacobian, then it's a bit more concrete. The point I was getting at about like attractor landscapes is more of it's like when you're in the domain of some numerical system that you can't give an eigenvalue, give some eigenvalues for, or at least in some cases you want to just simulate, and then kind of describe the phenomenology, and you can still like see when there's a behavior where you're kind of like moving around an attractor landscape numerically, right, and kind of describe that qualitatively, but not have some eigenvalues that you can kind of put a criterion on in that way. So yes, I agree, but I think it's kind of I think a general scenario. It's the same thing with bifurcations, really. It's like you have concrete mathematical definitions of bifurcation structure, and then you have the presence of bifurcations in the phenomenology of some system, like whether it's a simulated system or something you're observing empirically, and you often have particularly if you have some like interesting model where you can't write down what its higher order bifurcation structure is. You just need to describe the behavior you see. It's like, well, at this point it's got like low energy levels and zero oscillations, and then kind of move some order parameter up, and then clearly we get now like multiple attractors and bistability, whatever, like keep moving the bifurcation up, parameter up, and you see these regimes of different behavior types, right, numerically, but none of that is kind of something you can give you can't replicate that in a linearization, because it's something that's kind of way too numerically complicated and probably multivariable and nonlinear, and just wouldn't work. But it's still bifurcation, like in a kind of less mathematically tight definition of bifurcations, and so that kind of works, right? So I think it's a similar example there. Thank you, Ravindu. Good question. Any other questions from the room? Sharon. Okay. Sorry, may have been carried away. Eigenvalues remind you of zeta zeros because they have a real part that controls stability, eigenvalue controls oscillations. Yep. Yep, that's exactly what we're talking about. Great. Okay, great questions, great questions. Let's take a break. So we're at 3:30. So we'll do a 10-minute break. See you back here 3:40. And same as before, we have Colab notebooks. So browser, get ready for a link that'll appear in the Zoom chat. And we'll go through some practical examples of some of the stuff we've been hearing about.
Hello? Oh, hello. Can you guys hear me right now? Thumbs up in the chat if so.
Did you unmute on?
I did, yes.
Here's there, Sharon.
Okay,
Yeah.
perfect, perfect.
Sharon, I just introduced Baza. Baza, take it away.
All right, so guys, this would be, again, similar to the previous session, more of a hands-on implementation of what we just theoretically learned. This tutorial, as you might have guessed, is about the Wilson-Cowan equations. And so it's got 3 parts. The first part is we're just going to go over the basis of it, the framework of the model, how it's actually structured, just to see what the different parameters represent and how that influences the behavior of the system. And then the second part of it, we're actually going to be seeing the different ways through which we can actually look at the behavior of these equations, because at the end of the day we write these equations to get a behavior, and then that behavior is supposedly going to be interpreted in different applications, which brings us to the third part of the tutorial, which is actually the different applications that the different applications of this model in our real life cases, in actually like answering scientific questions and what kind of scientific questions we can pose and what we can actually do with the model. So without further ado, let's just get started. So let's quickly go through part 1, and as John described in much more detail, the structure of the model is essentially 2 different population types. One is excitatory and one is inhibitory. However, within each population type, we can have the 2 states of neurons. So they could be active or they could be quiescent. So the idea here is that they transition from active states into a quiescent state and vice versa. And so the other thing is that these populations of neurons are actually connected to each other. So the excitatory neurons feed into the inhibitory ones, and then the inhibitory ones feed back into the excitatory ones, and each population again is also connected to itself. And you can also have external inputs coming to each of the populations separately. So these are the parameters that the model has, the input parameters, the weights, the inputs, and the different populations. So now let's see what the actual equations are telling us. So this is essentially E of t and I of t are essentially giving us a representation of the proportion of active neurons. So this could be active of the excitatory population and active of the inhibitory population, E of t or I of t. But so the first differential equation here is saying that how the rates of active neurons are changing. So if they're active, and it's comprised, you can break it up into 2 parts, this part right here and then this part right here. So you can think of it this way. The assumption here is that the neurons that are active are going to decay with some probability rate after a while, right? And then the neurons that are inactive are going to become activated based on an input that comes into them, right? So if you look at the rate of change of the active neurons, the first part here is saying it's going to be negatively affected by the rate at which the active neurons become inactive. So the decay rate, right? So that's just saying that let's say like 70% are active right now, but after a while, it's going to go back down with some probability. So that's why we have a negative sign here. But then the factor that actually increases the probability, the proportion of active neurons, is this bit right here, which is saying of the inactive neurons, so 1 minus active, which gives you the proportion of inactive neurons. Of these inactive neurons, based on some probability and some input, they're going to become activated. So this input function right here is a sigmoid, and within the sigmoid we have the inputs to our into our populations. So this is just how it's structured. The so we got and these are pretty much symmetric for the excitatory and inhibitory populations. So if we just look at so if you just use these equations to couple these sorry. So over here, what we're just doing is looking at the behavior of these equations that we just defined based on the model parameters that we have. And just wanted to make sure that the different parameters are intuitive and we understand what each of them actually mean. So we have an alpha parameter over here for each of the populations, and the alpha is essentially telling us the decay rate, right? So let's just focus on the excitatory population right now. What do you guys think would happen to the rate of the active excitatory population if I increase alpha? And again, alpha is the decay rate. Yeah, exactly. It would go down. So like, if I just increase this, the rate of active neurons in the excitatory population would come down. So what if I change beta? If I increase beta, beta is again is the probability of a quiescent to active state. So if I just change beta, then we would assume that the percentage of the active population would increase. And if I turn it to 0, then we would expect it to decay decay to 0. So there's what this would mean if, for example, beta is 0, is saying that there is 0 probability that the quiescent neurons would activate and turn to active neurons. So if I just put it to 0, we only have the influence of the decaying active to quiescent neurons. But there's other, so for example, over here, if I change the behavior of the excitatory neurons, what I also see is that the inhibitory neurons activity also is changing. So what's up with that? Why is that? Yeah. So even though this is a parameter for the excitatory neurons, right? I'm changing it. And if you look at the yellow line over here, you can see that it's not extravagant, but you can see that the level of activity for the inhibitory population is also changing. So why is that? Why do you guys think that is? Sorry? Exactly. So if you look at the diagram, they're all connected to each other. So there is input from the excitatory to inhibitory, and that means that if the activity level of the excitatory changes, that also would influence the activity level of the inhibitory. Now, it's not a direct influence. That's why we don't see a dramatic shift, but that's the whole idea here, that these two populations are coupled together. So influencing one will lead to the change in the other, which would again lead to a change in the first population, and it has like that sort of like feedback interaction. And essentially that is what actually would allow us to and generate different dynamics. So here, there's a lot of different model parameters, which each of them influence the activity level of both populations. But that's not necessarily the interesting bit. There are a set of parameters if we put our model set up model with those parameters, we can see some different activities happening, some emergent activity happening from these two differential equations that we have. So for example, over here, this is very different from that steady state asymptotic convergence to a steady state that we were seeing before. So here we're seeing oscillations, right? So this is saying that these equations that we have can actually generate behaviors that we can associate with, we can use to describe, for example, neural behavior, the neural oscillations. So now the question is, well, we see that the equations can give us those types of behaviors, but now the question is, well, in what case, in what scenarios, how do you figure out from the equations what kind of behaviors these equations can give us? So that's kind of like, the point of using the equations to actually get some behavior out of them. And so that's where over here we are entering the second part of the tutorial, which is telling us how we actually can infer the behavior of these this dynamical system that we have, and different tools that we can use in order to do that, different methods that we can use. But before I proceed to the second part, just want to make sure that everyone is clear in the first part. Is there any questions? Yeah.
Yeah. Reset the link to the collab.
Yep. All right. Okay. So other than that, any other questions? Any conceptual questions? Is everyone good? All right. So now in this part, we're actually going to look at different ways that we can analyze the behavior of our system. There's two ways we can go about doing this, which are not, it's not exclusive, and they're more complementary than anything. But one is through an analytical method of just looking at the equations and seeing what the equations can tell us about the behavior of the system, which could be which would give us, well, I'll go through that, but the other method is just to simulate the equations, to actually let it play out and see through simulations, see how the system actually behaves. So one is just looking at the equations, inferring what it would do. The other one is to actually simulate it and see what it actually does. So let's see. So let's go through the analytical method here. So this is what we're getting into is looking at that's linear stability analysis, which is telling us that you just looking at the equations, we can see what the behavior of the system would be around a fixed point. So what do I mean by that? I'm just gonna quickly go over here, just show you the phase portrait of their dynamical systems. Essentially, the analytic method is telling us that if you have a fixed point in the system, then we can, from the equations, we can infer what the local activity of our dynamical system around that fixed point is going to be without necessarily having to simulate the data, right? And the way to actually go about doing that is, first, we have to find what the fixed point is. And to find the fixed point is essentially the confluence of the null lines of our system. So if you remember from the previous lecture, the null lines are regions in which the rate of change for our differential equations are 0. And the stable point is the point where both of them will become 0. So there is no movement in for either of the two dynamics involved. So the first step is actually doing that, is to find the stable point of our system by setting the equations to 0. And when we solve for that, both of these equations simultaneously, we will get a value for E and a value for I, which would then be the values at which we have the stable point. So then the next part of it is actually to take the Jacobian of our Jacobian matrix of our system at that specific stable point. So what the Jacobian is essentially telling us is a linearized version of how the system would change in each direction for either of the two dynamics. So we have a dynamic telling us about the rate of change of the excitatory population, and we have one for the inhibitory population. And then we have, so essentially the Jacobian is telling us for either of these activities, we take a partial derivative with respect to each of these two dimensions. So it's a two-dimensional system. We take the rate of change, the partial derivative of our excitatory equation with respect to E, same with again and again with respect to I, and then we do it again for the inhibitory population. So essentially, what the Jacobian is trying to tell us is the rate of change of either of the two dynamics with respect to either of the two dimensions that we have. And then once we have that matrix, then we can do eigen decomposition on that matrix, and that's how we actually get the eigenvalues for our the eigenvectors of these this matrix. And the eigenvalues, the components of the eigenvalue would actually give us allow us to infer the behavior of the system around that point. Specifically, if we have if there's a real component, if the real component of the eigenvalue is negative, that means that the system is going to be converging towards the stable point. If it's positive, that means it's going to be diverging outwards. And if it's negative, that means that it's not going to move. So one part of it is to look at the real value, and then the other part of it is to look at the imaginary component. If there's an imaginary component, then we would get a spiral effect. So that imaginary component would kind of signify a sort of like rotation. So this is the different combinations that you can get, for example. If over here, you can see that the system tends to, at the stable point, tends to diverge away. So what that would mean is that the real component is going to be positive, and there is going to be imaginary component to the eigenvalue. If there is no spiraling effect, then you can assume that the imaginary component is 0. So this is going to be the analytical method, but it's the advantage of it is that, well, it's going to be less computationally intensive because you don't need to simulate anything. However, it might prove challenging, which was actually the case over here, when you want to actually calculate and solve for the different the Jacobian matrix and taking the derivative, especially if, like, there's logarithms involved, which is the case over here in this example. So the other method of doing it is actually through numerical simulations. With numerical simulations, it's essentially just brute forcing it in a way, looking at generating a phase portrait by looking at the rate of change at every possible point in the grid, every possible location, and then also initializing your system in a initial state, and then seeing how that initial state will evolve by essentially solving for the putting the initial system into your equations and taking a stepwise approach in solving the equations and seeing how that actually evolves, that system evolves. So this over here, you just need to be, simulating data and seeing how the system would behave. All right. Any questions so far about? I know this might have been a lot, especially for people who don't have experience with. Yep.
Is it, do you see cases where the complex plot is 0? Like, do we have to see a case where we have to get some point? It's not like just either.
Yeah, that's a great question, actually. So a saddle point, if you think about it, what it means is that there's a convergence in one dimension, but a divergence in another dimension. So so it's like, kind of like sloping down and like coming in, right? Well, the saddle points, yeah, the complex, it has to be 0. Yeah, I think so. Yeah. Well, yeah. If the complex part is 0, that wouldn't necessarily tell you if there's a saddle point or not. It would just tell you if it's a spiral or not. If it's kind of like, spiraling inwards or outwards, or if it's just a straight line converging into a node. In the case of a saddle node, it actually depends on if the eigenvectors have opposite signs. So we have a Jacobian matrix, which is a two-dimensional matrix, right? So if we take the eigenvalues of it, we get two eigenvalues, right? So in each we get two eigenvectors, and each eigenvector has its own set of eigenvalues, right? So if one is positive and one is negative, that's where I actually get the saddle point. So the conditions for having a saddle point is actually not necessarily due to the imaginary component, but the real components of either of the eigenvalues having different signs, if that makes sense. Right, right. Any other?
I'm not sure if this was, maybe you're getting at this part of this question, but you're saying like, do you ever see that in practice? So that could mean a few things. One version of it is that something that's kind of studied a lot in physics literature. And I think it's the case that the Lefebvre model, which is not this model, but it's like somewhat related, or maybe it's more similar to the Morris Leclerc, but it's a widely used model for epilepsy, particularly the kind of movement into and outside of seizures as being bifurcation, in entering and exiting bifurcation. So that it's a five-dimensional system that has like 2 bifurcations and all the theory on it, and but there's regimes where you have like presence of a saddle, presence of a stable mode, presence of an unstable focus in parameter regimes that are supposed to replicate the phenomenology of seizures, like stimulation of seizure traces. But there are like real world examples of some of those scenarios, parts of stuff right there. We'll also count the properties as well, but I can't tell stuff in my head. But if you want to find like somewhat, you know, hands-on example of something that, so it's like Epileptor, EPILEPTR. I think we might touch on that in one of the last lectures. If you like your dynamical systems, then you'll probably like Epileptor. It's sort of like
I think there's another question in the chat. Hi, Parsa. So when we talk about stability here, it's strictly local, yes? Or are there methods using some well-defined Lyapunov functions that can be used to show these fixed points are globally stable? So yeah, when you're looking at the behavior, yeah, you're looking at a local kind of region within your phase portrait. That's why you need to define first a point at which you're looking at the behavior. So what we're doing here is looking at the stable points, the behavior around the stable points. And the second part of the question is, are there methods using some well-defined Lyapunov functions that can be used to show these fixed points are globally stable? There are, I'm not sure if there's a systematic way of doing it, but you can infer the overall stability by just taking the Lyapunov exponents at different regions of the phase portrait. I don't remember if it on the top of my head, but that would kind of like let you categorize if the system is going to be stable, quasi-stable. I think chaotic. You can also tell if it's a chaotic system or not. I'm not exactly sure. I think there are some methods, but I'm not sure in the top of my head. Sorry. Yeah.
Yeah, comment slash question that couldn't you just see the actual values, like the eigenvalues that it kind of spits out, like the equilibrium solutions and that kind of phase portrait, and then just see, okay, my phase portrait has 2 spirals, 1 saddle point, whatever, and then like you got like the argument of, do you need some sort of other function? Isn't it kind of just defined by like how we set up linearized differential equations that depending on where you just like choose your initial condition, it will just go to one of those points and or go crazy, whatever. Like I think she's asking about like
scenario without doing the local linearization.
Oh, but we're not doing that right now, right? Okay, okay. Just curious, because
I think, Sharon,
It's urban do, yes.
I know kind of generally speaking, the kind of thing you're talking about, but I've never really seen, I don't see that come up in the kind of literature on these kind of models in the neuroscience context. I think
Right.
the reason, broadly speaking, is you know, even there might be some global analysis on epilepsy actually, but like broadly speaking, like what we're doing here is we're doing some real kind of head down simplification of the dynamics of the model that ultimately we want to be describing a rather more complicated set of things that we're measuring in neural activity. So the local linearization is quite a high level of abstraction relative to the experimental data that it's describing. Does a decent job, but there's a lot of abstraction and kind of simplification there. I think the context where you can give some global conditions for certain dynamics behaviors is moving outside of any real practical relationship to like measurable data, and at that point it's like only a mathematical exercise, which is, you know, there's lots of work on that, but most of the, say, action in computational neuroscience is, you know, trying to kind of hit that sweet spot where we can define a, you know, simplified, abstracted, like locally linearized system, and then have that retain something that we can connect to some neuroscience data. And I think the cases where you can give global conditions for some dynamical properties are very boutique.
Right. So you're saying at this point of the analysis, finding global conditions is actually not going to contribute to the analysis.
Yeah, I think
Right.
if you're talking about like phenomena that you like that are scientifically interesting, rather than kind of just mathematically interesting, I don't think there's much that comes from trying to find global conditions on these kind of.
I understand.
It might be
Right.
the case for some of the bifurcation things in epilepsy, though. There's like conditions for codimension 2 bifurcations that I guess is like global. But I'm broadly speaking, yeah, the local linearization is as far as your kind of simplifications are going to go and still be useful.
Right. Understood. Thank you.
Thanks.
I do have a question about, I think it's about a question about just the, what global, like, I guess convergence equilibrium actually implies. It's not implying that if I look at my face portrait and select some sort of an initial condition, that it will always go to one specific equilibrium. Is that kind of what that sort of did? Because like, okay, yeah, because that kind of just doesn't make sense. Because if you have like, if you do your Jacobian and then you have a whole bunch of different equilibrium solutions, could be
Yeah.
a spiral, could be a source, whatever, depending on your initial condition, it will
Fall into
it will
different
fall
regime.
to one of the several equilibrium solutions. There would be no such thing as a global.
Well-
Like to say that it at any mean on the face portrait, it would go to one specific equilibrium solution. Like you could
It would be a
construct
very
that.
dumb system.
Yeah, you could construct that figuratively,
Yeah.
but I mean, like, I don't know. Like, I'm even.
Yeah, I'm
looking
not
further down.
sure what kind of like dynamically interesting system
Yeah.
would accommodate that, but like that's.
Like further down, I couldn't imagine that, you know, like they might have different solutions depending. Also reference to kind of what he was saying before.
Right.
You could argue that this also might be a saddle point. We don't know what it looks like, so he was curious as to where it shows up. Maybe even in our, you know, the rest of the tutorial. But
Oh, there's definitely saddle points for these systems, yeah.
yeah,
And you know, you can kind of like identify if there's going to be saddle points, or if there's going to be stable points, just by seeing if there's solutions to setting the derivatives to 0 and seeing like how many solutions you can get. And if I remember correctly, so just by noticing, by seeing you can kind of like go the other way around as well. So over here in our face portrait, we're seeing that we get a, for example, a limit cycle here, but we only find one fixed point. But I'm not exactly sure, so I'm not too confident in saying this. But in the instances where we had a limit cycle, I think it has to be the case that we would require another stable point with the opposite sign to exist to kind of like, you know, counterbalance the if this is pushing it away, something has to be pulling it back in. So there might be another stable point outside of the domain that we're looking at it. Right, yeah. But again, I'm not too confident in saying that.
Excuse me. So, Professor, you're saying that this system is sensitive to initial conditions?
Well, that would be a different description. That would be if you're talking about sensitivity to initial conditions in like chaotic systems, that's somewhat different. Here the system isn't necessarily, the system as a whole isn't sensitive to initial conditions. It's what is sensitive to initial conditions is the evolution of this particular instance based on the initial conditions. So for example, it might be the case that we have another stable point over here, another stability domain over here, that if you initialize your system over here, will just fall into that in that basin of attraction. But it just so happens like a bistable system. But it just so happens that because we initialize over here, the initial conditions over here, it wouldn't get to reach that other attractive state. It would fall into this kind of attractive regime. So it's the system itself isn't sensitive to initial conditions. It's the particular evolution of what you're simulating that would be potentially sensitive to initial conditions, if that makes sense.
Yeah, thank you. Yeah.
No worries. Okay, so yeah, so over here we're going to be looking at the numerical method, and the numerical method is pretty straightforward. You just generate the phase portrait by you have the equations for rate of change depending on different values of E and I, and that would essentially give you these arrows. If you just solve for different points in this phase portrait, then you can generate the kind of vector field. And then when you initialize a specific point, at a specific point, you can see that your system falls into how your system evolves just numerically by simulating it. And there are instances where you would actually want some sort of like somewhat of a noisy to incorporate noise into your system. So what we have here is a strictly deterministic system, but that might not necessarily be biologically match biological recording. So you want to kind of like sometimes you want to inject a sort of noise into the system as well, which is what you're seeing in this right-hand side of the equation, or right-hand side of the plot. So actually I do have, I think, yeah, this is interactive. So for example, if you have a noisy system as well, there's another way you can get oscillations, which John briefly mentioned. For example, over here, we're saying that the deterministic version, so this is kind of like the same, these are the same systems, only the right side has that noisy integration step added in to simulate some sort of fluctuations. But for example, if we have a case where, let's see.
This little bit on the right.
Yep.
Because it doesn't look that noisy.
Yes. So I'm just gonna amp up the standard deviation of the noise. There you go. So it's gonna be much easier. And you can see instances where you would get oscillations strictly because of the injection of the noise. So over here, let's let it load. Maybe the noise is too much here, but you can see in the deterministic instance, there's just a stable node. or say stable spiral, where the system falls into that stable point and it doesn't change anymore. But if you inject noise into the system, or you have a noisy trajectory, it will give you that sort of sense of oscillation, which is essentially when the system is at the stable point, it just shifts it again a little bit, perturbs it a little bit farther away from the stable point, which causes it to converge back, which then kicks it out again and causes it to converge back again. So you might also get some sort of oscillations just through the effect of noise. So over here, any questions about numerical integration and the numerical, just simulation method so far? Okay, so there's an exercise that you guys have to do, and this is I think this would be probably the most extended exercise so far that you guys have done, which is essentially to do linear stability analysis in a using both methods, using kind of like a combination of both methods. So what I want you to do, well, first of all, I want you guys to understand what's going on so that you will be able to do it. But what specifically you guys are supposed to be doing in this example for this exercise is to set up a combination of parameters that's at your own choosing, whatever you find interesting, and find the stable point. And through numerical integration, you would be able to actually find the location where the stable point is occurring. And then once you have the coordinates for the stable points, you guys are going to go back and calculate the Jacobian matrix of it, and then do the steps for inferring the stability of it. Get the Jacobian matrix, do a decomposition on the Jacobian matrix, and see if the components of the eigenvalues, the real part and the imaginary part, if the behavior that you would infer from these components match the what you're observing in the numerical method. Is that clear? Is that clear what the objective of the exercise is? Or would you guys want me to go over it again? Okay. I think there's another question.
Do you want to break for 5 minutes so we can work on this, or 10 minutes?
Oh, this is a take-home assignment. I don't think.
Okay.
Yeah. The graph for eigenvalues and stability is very interesting. Is there an extended or more continuous as opposed to snapshot graph of this, or more dynamic animated visualization? So okay, so I'm guessing you're talking about this one. So it might have been before we introduced this plot. So this plot will be more
one above it, the black one. If you go scroll up more.
over
The-
here.
Yes.
Oh yeah, this is just a slide that I screenshotted from the lecture slides. But you might be able to generate the same sort of behaviors by playing around with the parameters of the systems that we have so far. So for example, we can see that we can transition between a limit cycle and a stable spiral by moving around the parameters. So over here, what you saw was a stable spiral, but the initial with the parameters that's initialized by default, you see a limit cycle. So I don't have any specific example that would generate these type of plots for you, but you can try to replicate these different scenarios by trying to manipulate the different parameters of your system. I don't know if that answers your question, Shireen.
Yes, I was just wondering if these oscillations can be seen in action, like as they oscillate.
Right.
Like a
Yeah.
visual animation of the whole process, not just like a one moment of it.
It's supposed to be kind of like capturing that sense of progression through a snapshot. So that's kind of like a useful value of having face portraits. Even though it's like a static representation, within that static representation, it gives you that the information that you would need to infer what it would look like dynamically. So for example, the direction of the arrows would tell you how it would be, how the system would be changing. So I guess like a visualization of it is just for you to imagine these arrows keep moving on these and these lines that you're seeing.
I think if you go to the phase plane plots that you've done, they're both the trajectories, right? So, Shireen, like, are you kind of clear how these are showing the evolution over time?
Yes.
Yeah, okay. So you were saying like, do we have any examples of these trajectories for the example figures from the picture above?
Yeah, no, I wasn't really looking at these two together. I was just wondering, those patterns, that shape and the spiral or the lines. It would be interesting to see, if these coordinates were moving and as the brain works, I don't know how to put it, but I was just wondering, is a more like animated version of this visualization? Maybe that doesn't make sense as a question.
No, it's a great question. I think the answer to it, I think partly is the figure that you've got in front of you. So it's on the slide right now. So if you scroll down a bit more further, so you see both of them. So one of the ways that these are often shown, which makes it very clear the relationship between them, is to kind of roll out the trajectory and the phase plane on the top. So say the top right, and the time series on the bottom right. And what you'd see if you did those like as a movie, as you're suggesting, is you kind of have some starting point for the trajectory on the top right, and then it would just basically cycle round, right? So you kind of draw out those green lines or the black lines on the left, right? And as those lines get drawn out, like every loop is one of the periods of the oscillation, right? So you can see the evolution in the time series plots because there's time, but for the phase plane plots, it can be useful to have a movie because there isn't any time dimension on the phase plane plots, right? Like the thing you're seeing is it's kind of squashed through time in the traces. But if you see if you plot the traces like as a movie, then you kind of see a little bit more clearly how the trajectories in the phase plane correspond to, an oscillation over time. Does that help?
Exactly. I was wondering if like there's something like that on YouTube or somewhere you've seen that you can, if it's been.
Yeah, I mean, we could do it with this code. Like it'd be kind of straightforward to just like generate a movie, like a set of frames, like in this notebook. That would be a fun exercise, actually. It's pretty easy to do with the structure in these notebooks to kind of generate some visual movies that kind of maybe demonstrate these things a little bit more
Great,
dynamic.
I'll have a go.
Yeah, the way to do it, the way I recommend for making movies is MoviePy, and you just save a bunch of PNGs and then use MoviePy to just combine a bunch of PNGs. There's other ways to make movies in Python, and they always break in my experience. MoviePy is the robust one.
And I was thinking React, but I'll have a look at it. Thank you.
Just I'll offer a resource. Actually, I'm going to send it into the link chat. It's just a phase portrait plotter that I was introduced to my like ODS class in the past, and it kind of hopefully does what you're kind of looking for, where you can just kind of select any initial condition and it kind of animates the kind of flow of the phase portrait. So I think it's like that was something that helped me a lot to help like visualize and comprehend how phase portraits just the dynamics.
Okay, cool. Thanks for that.
Yeah.
Yeah. Okay, so, and everyone is comfortable with the exercise. Everyone knows what we want for the exercise. Okay,
Yeah, so to be clear for the exercise, guys, I think Paz is wrapping up, so I'll just give a comment on the exercises as well. So we've come to the end of week 3. Actually, a little round of applause for Paz.
well, there's one more section remaining, just one more part 3, but
Well,
like,
let's come back. You finish and then
okay,
come back to the exercise question. So I
Okay.
want to just communicate that like over the last 3 because they cover as a group. But we'll finish this up and then I'll give that.
okay, gotcha. So the last part of the tutorial is actually, now that we have understood the structure of the model, and now that we've understood different ways that we can analyze the behavior of it, what can we use the model for? In what cases we can actually use the model? So these are some of the use cases that the model would be beneficial for. For example, you can generate synthetic data based on the models that's supposed to represent, some activity of the brain, some sort of oscillations. All right, you can generate synthetic data, and then you can see, for example, which parameters would influence the which parameters would be sensitive to the data that you simulate. Other ways you can actually use it is to kind of like have a hypothesis of, for example, this is what's going on in the brain, and I don't have access directly to the brain, what's going on inside the brain, but I have this proxy model. And what I can do is tune the parameters of this model such that it actually reproduces what I'm observing empirically. And then I can infer what's going on empirically by just looking at the parameters of the model with the synthetic data. So anyway, you can read through them on your own time. 2 examples that I wanted to show is that, for example, by simulating the data over just an extended period, you can actually generate raster plots. We had something similar in the lecture slides, which is just having a bunch of like a large number of neurons and using the Wilson-Cowan equations to synthetically generate spiking data, which we won't get into the specifics of. But then once you have the spiking data, you can compare it to an empirically recorded spiking diagrams or raster plots. And another use case, for example, would be to generate the time series, and from the time series, you can actually generate a power spectrum. And this is the same way that you would actually generate power spectrum from empirical time series. You can just use synthetic time series and we get power spectra in that way as well. So there's different use cases for your models as well, depending on your question, depending what you want to be looking at. And yeah, so that wraps up the tutorial. And if there are any questions, would be more than happy to answer. If not, well, yeah, thank you for your attention.
Okay, so I'll just make my comment now. So this brings us this concludes week 3 of course. What's coming up next week, if people have taken a look at the schedule, is nothing. So there's a break in the schedule. The reason for the break is because I'm not here, but it's also I think a good pause point. The assignments structure for the graded part of the course, as you recall, are assignment 1 and assignment 2. And as we discussed earlier, so both of those are going to be essentially like all of the exercises for the first 3 and then the next few lectures. So what your job is, and the submission time for this is actually it's basically the end of the course, so you don't need to do this right now. But what we will do in the next few days is we'll just drop a note on the Moodle, kind of saying very clearly that assignment 1 is released like officially, and what that will consist of is some additional links. So the one we haven't given the links for the ones with the assignments.
Well, no, everyone,
Okay.
all of the assignments, all the questions to the assignment is already, you already have them. They're in the, yeah,
So
exactly.
So you already have this. They're at the end of each of the co-op notebooks.
Or in the middle, in this case, it's in the middle.
Okay.
But you will be able to identify, it clearly says exercise or
We're
assignment.
saying this is the assignment, so there's like a few little pieces of the full assignment in the 3 notebooks for the 3 lectures that we've done so far. And so assignment 1 will be to just do each of those, and then what we'll ask for is for you to give us the notebooks, right?
Just the answers to them.
Just the answer.
Yeah, because it's like there's 3 different notebooks. You can fill it out within the notebook and upload the notebook, or you can just, you know, put it in a separate document. But essentially
So we'll give some specs on what we want the submission structure to be, but we'll kind of summarize what the composition of those things is. You have materials. If you want to get on with that, you know, in the kind of off week, then that might be a wise thing to spend some time on if you want to kind of stay fresh. But otherwise, like, you can do that at any point until week 9 or so, which is when we want to have
things wrapped up.
done.
Things wrapped up.
I think doing assignment 1 sooner rather than later would
Yeah.
be a good idea for you and a good idea for us. But we don't have a hard date on the submission for assignment 1. But yeah, you can get started on it fairly soon. And it's covering stuff we've seen in the last 3 lectures. So see you guys in 2 weeks. And as I've said previously, keep the discussions going on the Moodle. I mean, especially with the assignments coming out now, that might be a good time to kind of reflect on some of the stuff that you were thinking about, and, you know, drop some questions, drop some ideas, share some insights, whatever occurs to you. And yeah, enjoy. Okay. So see you guys in 2 weeks.